Now, iodine test is the type of test which is used for detecting certain type of sugar, especially starch. Starch is a polysaccharide. We have learned about it. It's a homopolysaccharide. So starch is a type of test which detects the presence of starch. If you have a sample, you want to test that if there is starch or not inside the sample, then you do the iodine test. So we can say iodine test is positive. When iodine test is positive, it means that the sample contains starch. Okay, so it mainly tells if starch is present or not. Now, with every carbohydrate with a different sugar, it gives different type of colors. Let's start with starch itself, and then let's test a sample with glycogen and cellulose, because these three are the most important polysaccharides. All of them are homopolysaccharides, which means that they have the same type of monomer repeating units. Starch has alpha glucose in it, cellulose has beta glucose, while glycogen has alpha glucose. But the difference between starch and glycogen is that starch is branched and unbranched, which means it has two units, amylose and amylopectin, while glycogen only has branched chains. Okay, so there is difference of bond formation. I mean, the bond here is 1,4 and 1,6, but in glycogen it's just 1,6, alpha 1,6 glycosidic bond. Okay, anyway, so that was it. This was revision of what we have done in the past about the sugars. Clear? Now, which sugar will give what color with iodine? So let's start with starch. If you get blue black sort of color, it means that there is starch in the sample. Okay, so starch gives blue black color in terms of like definitely iodine test we were talking about. When it comes to glycogen, so it gives like a less impressive color, that is reddish brown. Okay, you need to remember that what color comes up with a different type of sugar. And cellulose don't really give any color. Okay, no color change will happen in cellulose. Fine. So here the right answer would be all monosaccharides give positive iodine test. No, because iodine test is mainly for detecting starch, and all disaccharides, no. Only starch gives a positive test for iodine. Amylose, now amylose is a component of starch. Okay, so definitely it's going to give positive iodine test. Cellulose don't really have any color, no color change, so it's a negative iodine test. Glycogen only no. So cellulose is going to be the right, amylose is going to be the right option, because amylose is a component of starch. Okay, let's talk about how many molecules of ATP is going to be produced by using one molecule of glucose via cellular respiration. Cellular respiration is a process. It's a catabolic process in which you break down one molecule of glucose. Just for reference, one molecule, we are going to talk about it. If you break down one molecule of glucose in the presence of oxygen, then it's going to yield, means give how many ATP approximately. So in eukaryotic cell, approximately you're going to get about 36 ATP by one glucose, and in prokaryotic cells it's going to give you 38 ATP in prokaryotic cell. Cellular respiration. Why there is two more ATP here? The simple reason behind it, just to be very short, prokaryotes don't really have any organelles, so everything is going to happen in the cytoplasm. So the molecules don't really have to go inside any other organelle. and thus it's not going to use up any energy. But in eukaryotes, when pyruvate enters mitochondria, so it uses up 2 ATP. So that is why it makes 38 in total, but then you use 2 ATP to make pyruvate go inside mitochondria. That is why we say it, the net profit is of 36 ATP in eukaryotes. That's, and then the ideal number of ATPs that are going to be produced by one molecule of glucose. Now if you look at the options, this is the most closest option that we have. That is why option C is the correct option. Okay, let's talk about question 39. Raffinose, a trisaccharide. It's found in beans, and you have to name the monosaccharides. Now this is a not very common sugar, so now that you have heard about it, so remember that there is glucose, fructose, and galactose inside raffinose. These are its monomers or monosaccharides. Okay, but it's not a very common sugar. Okay, so I hope that is clear. Let's move to the next. In this question, it's asking the conversion of glucose into 2 pyruvate molecules during glycolysis involves. So when you break down glucose in cellular respiration, there are 3 steps. Number 1, glycolysis, where glucose is broken down into 2 molecules of pyruvate. Okay, so you form 2 molecules of pyruvate, and then these pyruvates enter mitochondrial matrix, where step number 2, Krebs cycle happen. So glycolysis is happening in the cytoplasm, Krebs cycle is happening in the mitochondrial matrix, and in the 3rd step, it's electron transport chain, which happens in the inner mitochondrial membrane, which is also called as crista. So this is electron transport chain or oxidative chain, where you produce more ATP. Now it's a long process we are going to talk about in the chapter bioenergetics, but here the question asks that if you break down glucose into pyruvate molecules during glycolysis, yes, that's what we do in glycolysis. So first of all, it's a catabolic process, it's not anabolic process. Okay, it's not anabolic process because you're not really making something, but you're breaking down glucose into pyruvate, and then you are releasing energy in the form of ATP. And the ATP which is formed by glycolysis, that ATP is called as substrate level ATP, and the process of making that ATP is called as substrate level phosphorylation. Phosphorylation means addition of phosphate. So when you add phosphate to ATP, you form ATP. So whenever you look at this word, when you hear this word, remember that it's about formation of ATP. So if you form ATP in glycolysis, it's by substrate level phosphorylation. If you form ATP in electron transport chain, it's called as oxidative phosphorylation. If you form ATP in photosynthesis during light reaction, that is called as photophosphorylation, because photo means light, so you're forming ATP using light. So these are different types of formation of ATP. So substrate level means that you are not actually using any catalyst here, but you are just forming ATP by the substrate is a molecule which actually combines with the enzyme, and enzyme acts on the substrate and form a new compound. So in substrate level phosphorylation, a group is attached to, you're in simple words, you are forming the ATP using the substrate. So glycolysis is a 10-step process, right? The first 5 steps you are using ATP, and then in the last 5 steps of glycolysis you are generating ATP. So in glycolysis you get a net profit of 2 ATP, and these ATPs are formed by substrate level phosphorylation, because in these 10 steps you are forming different sorts of compounds. We have different sorts of molecules which are formed one by one, so there, that way we have 10 steps. So during these 10 steps you have like different compounds, different 10 compounds here. So if we are getting the phosphate group from one of those substrates, one of those 10 compounds, and then you are adding that phosphate to ADP, and that's how you're forming the ATP, that's called as substrate level phosphorylation. You don't need any oxygen. So glycolysis is, it happens in the absence of oxygen. It can happen in the absence of oxygen. You don't need oxygen, so it actually happens in the absence of oxygen, right? So if you look at the options, it's not condensation reactions only. There is catabolic reactions too. So condensation means forming new bonds, formation of bonds by removal of H2O. So that's incorrect. If you look at option C, that's partial oxidation of glucose. So the whole process of cellular respiration, it's oxidation of glucose or breakdown of glucose. So in every step you are doing some oxidation of glucose. In glycolysis it was not complete oxidation, because if you do complete oxidation in the first step, then there is no need of doing Krebs cycle and electron transport chain. So glycolysis is actually, indeed, it is partial oxidation of glucose, and then in Krebs cycle it's partial oxidation, and in electron transport chain, that's where the complete oxidation of glucose happen. And then in glycolysis you form ATP. That's what I just explained you here, that it happens because of substrate level phosphorylation. Option D says complete oxidation to carbon dioxide. That's not true. It's not complete oxidation, it's partial oxidation. Hydrolysis of glycogen directly. So that's not what we do in glycolysis. You break down glucose, not glycogen. So option C is the most appropriate option here.